Welcome to medicinal biochemistry class on protein and amino acid metabolism. So today we will learn about catabolism of amino acids. So how these amino acids are broken down. So what are the reactions that are involved in catabolism of amino acids? Okay. So at the end of this class, the student shall be able to explain transamination reaction, recall significance of transamination reaction, explain deamination reactions, recall significance of deamination reaction, explain decarboxylation reactions, and recall significance of decarboxylation reactions. Okay. So there are three major catabolic reactions of amino acids. So that is transamination, deamination, and decarboxylation. Okay. So transamination, so the name itself suggests what will be happening in that. So here, so the amino acid transfers its amino group to one more molecule. So here the other molecule is an keto acid. So here you have an alpha amino acid. So here is the amino group, and here is the keto acid. So it contains ketonic functional group as well as carboxylic acid functional group. So it will transfer this amino group to alpha ketoglutarate, and alpha ketoglutarate gets converted to an amino acid, that is glutamate. So amino group is added over here at the second carbon atom, and the amino acid gets converted to alpha keto acid. So the keto acid gets in transamination reaction, the keto acid gets converted to an amino acid, and the amino acid gets converted to keto acid. So the reaction is catalyzed by aminotransferases, and it requires pyridoxal phosphate as cofactor. So that is, so in summary, so amino group from an amino acid is transferred to a keto acid. So a new amino acid and a new keto acid is formed. So this is catalyzed by a group of enzymes called as transaminases or aminotransferases, and it requires pyridoxal phosphate as cofactor, and a specific transaminase catalyze each pair of amino acid and keto acid. Okay. So aspartate transaminase and alanine transaminase are two important transaminases. So they make significant contribution to happiness. Okay. So transamination is reversible, hence it involves both anabolism and catabolism. Okay. So the significance, when coming to its pharmacological significance or biological significance, liver, kidney, heart, brain have adequate amount of these enzymes, and transamination is important for redistribution of amino groups, production of non-essential amino acids, and for the production of non-essential amino acids. So it diverts excess of amino acids towards energy generation, and that is for gluconeogenesis. And amino acids undergo transamination to finally concentrate nitrogen in glutamate, and this glutamate will undergo oxidative deamination to liberate free ammonia for urea synthesis. Okay, all amino acids except lysine, threonine, proline, and hydroxyproline participate in transamination reaction. Okay, so the enzymes that are present within the cell are released in the cellular during the cellular damage into the blood. Okay, unless there is any damage, you won't see certain enzymes, mainly AST and ALT. So AST is increased, the level of AST is increased during myocardial infarction, and AST and ALT level increases in case of hepatitis and alcoholic cirrhosis. So this measurement of these enzymes will help us to diagnose certain diseases. So this also happens during muscular dystrophy, breakdown of proteins. Okay. So the next thing is deamination. Deamination, the name itself suggests that is a removal of amino group from amino acid. Okay, so deamination is of two types: oxidative deamination and non-oxidative deamination. Oxidative deamination is the liberation of free ammonia from amino group, that is from the amino group of amino acid. So this is coupled with oxidation. So this happens mostly in liver and kidney. And oxidative deamination, the role of oxidative deamination is to provide ammonia for urea synthesis and alpha keto acid for a wide variety of reaction, including energy generation. Okay. Oxidative deamination is carried out by three major enzymes: L-amino acid oxidase, D-amino acid oxidase, and glutamate dehydrogenase. Okay, so the oxidative deamination of L-amino acid oxidase. So here L-amino acid oxidase mainly catalyzes the conversion, deamination of L-amino acids. So this L denotes the position of that is a spatial arrangement of ammonia NH2 group. So this catalyzes specifically L-amino acids. So this will remove the L-amino group from L-amino acid and it will convert it into alpha keto acid. So here the reducing equivalents are taken up by the FMN and they are converted to FMNH2. Okay. And amino acid gets converted to amino acid and then to keto acid. So this enzyme is present in liver and kidney. So the activity is very low. It requires FMN as a cofactor and it deaminates naturally occurring L-amino acid. This enzyme deaminates naturally occurring. So the next thing is oxidative deamination by D-amino acid oxidase. So this specifically deaminates the amino acid with D configuration. And so here previously it required FMN and here it requires FAD as cofactor which gets converted to FADH2 and it forms alpha amino acid and alpha keto acid. Okay, so this is present in plant and bacterial cell wall, require FAD as coenzyme and deaminates naturally occurring L-amino acids. Okay. So the next is oxidative deamination by glutamate dehydrogenase. So here you have glutamate. Glutamate takes up by the action of glutamate dehydrogenase. It loses, it transfers its reducing equivalent to NADP, and NADP is converted to NADPH, and this gets converted to alpha aminoglutarate, which again is converted to alpha keto glutamate. Okay. Glutamate rapidly undergoes oxidative deamination catalyzed by glutamate dehydrogenase to liberate free ammonia. So it can utilize NAD or NADP as a coenzyme. So glutamate dehydrogenase is involved in both anabolic and catabolic because the reaction is reversible. So amino groups of most amino acids are transferred to alpha ketoglutarate to produce glutamate. Glutamate is the end product of amino acids which ultimately releases the ammonia group to form urea. Okay, so the glutamate serves as a collection center for amino acids in biological system. It reversibly links glutamate metabolism with TCA cycle through alpha ketoglutarate. Okay, so non-oxidative deamination. So deamination of amino acids to liberate ammonia without undergoing oxidation is called as non-oxidative deamination. So this is catalyzed by two enzymes, dehydratases and desulfurases. So hydroxyamino acids like serine, threonine, homoserine undergo non-oxidative deamination catalyzed by PLP dependent dehydratases or dehydratases. Okay, so here you have the example: serine. Serine is converted to, it loses a molecule of water, it gets converted to amino acrylate, and serine dehydratase will convert amino acrylate to pyruvic acid. So here the amino group is thymine. Okay. So non-oxidative deamination by amino acid desulfurases. Sulfur-containing amino acids, mainly cysteine, homocysteine, undergo deamination coupled with desulfuration to give keto acids. So cysteine is converted to alpha amino acid by the loss of H2S gas. So here you require dehydratase enzyme, dehydrase enzyme, and PLP as cofactor. And alpha amino acid is converted to pyruvic acid by the action of desulfurase. Okay, so then finally decarboxylation. Decarboxylation is removal of carbon dioxide from amino acid with formation of amines. Okay, so here you have the general structure of amino acid. You are going to remove carbon dioxide. So here a simple amine is formed. So what is the significance of decarboxylation? Decarboxylation helps in formation of physiologically active compounds. For example, GABA. GABA is synthesized from glutamate. So glutamate dehydrogenase is the enzyme that is required. GABA is a neurotransmitter. Then tryptophan is converted to serotonin, so that this serotonin stabilizes our mood, feelings of well-being, and It helps us to keep us happy. So it helps in sleeping, eating, and digestion. So that is why serotonin is a very important biological molecule. So catabolism of amino acids during decomposition of protein. It helps in catabolism of amino acids during protein decomposition. So the enzymes of microorganisms that are present in colon dead organism, they remove carbon dioxide from amino acid with the formation of diamines. Ornithine is converted to putrescine. Lysine is converted to cadaverine. So this is about the catabolic reactions of amino acids. So in summary, the transamination reaction, so there are three major reactions: transamination, and deamination, and decarboxylation. The transamination reaction, amino group is from an amino acid is transferred to keto acid. A new amino acid and keto acid is formed, and enzymes that are present within the cell are released during the cellular damage. Okay, like alanine transaminase and aspartate transaminase. So they are released during myocardial infarction and hepatic and alcoholic cirrhosis. Hepatitis and alcoholic cirrhosis. So the removal of amino group from amino acids is known as deamination. So there are two different types: oxidative and non-oxidative deamination, bring energy generation. Okay. So the role of deamination is to provide free ammonia for urea synthesis and also alpha keto acids for variety of reactions, including the energy generation. Okay. So the removal of carbon dioxide from amino acid results in with the formation of amines. So this is decarboxylation reaction. Physiologically active compounds are synthesized by decarboxylation, and decarboxylation can lead to decomposition of proteins. Okay, so this is again Bloom's triangle. So this explains different levels of learning. Remember, recognizing and recalling the facts is called as remember. Understanding what facts mean. So that is understanding. So you should understand better to remember better. Then apply the facts, rules and concepts and ideas that help you to master the topic. So then breaking down the information into component parts. If you are having a larger problem, break down and study the problem and try to resolve it. So if you have achieved these 4 stages through hard work and persistence, then judging the value of information or ideas, you can do it, and combining these various parts of your learning into a new whole will give you the new dimension towards your work. Okay, so finally, happy learning. Thank you.